PhD from Bose Institute, Kolkata, and then he went to Germany as a humble fellow, and then worked with David Eisenberg as a very eminent scientist in his postdoc, going to USA, and then he joined IIC Bangalore. He has recipient of many of the very renowned and famous uh, degrees and other things. So he is working in the computational data science department. During his PhD, he worked with protein folding, protein structures, isolations, and actually he is a true bioinformatician and he knows the program very well. So he is a person who worked with the not only with the protein data set, protein data bank, he worked also with different databases also. So I think it is a good opportunity for you who will take you from database to the big data and few applications of AI, which is a demand of the future biology. So with this little introduction, I will request Professor Paul to take over the mic and continue. Professor Paul, please.
Okay. Good evening, all. Am I audible?
Yes, sir.
Okay. So, do we need the video by any chance, or we can just be anonymous? Okay.
Recording in progress.
So we will start. Uh, we will start by a quick introduction. So myself, Devnath, currently working in Institute of Science, Bangalore, and my area of work is computational biology. So before I move on, I just want to understand how many persons in the audience have a background in computer science and how many persons have a background in biology. So that will help me emphasize certain aspects more and bring clarity. If you're from computer science background, maybe you can just give a shout so that I can quickly get to know that you're from computer science.
Myself, Bal, I'm actually from computer science and information technology department.
Okay.
Working as assistant professor.
Okay, I have one person. Any other person from computer science? Okay, so I understand all others are from biology, right? Or maybe chemistry. Okay, good. So let's get started. So today, our goal is to introduce you to database in biology. And since this is a faculty development program, the idea is to give you new, topics that you may want to uh, teach, or you may want to, engage yourself in some kind of a small project. And computational biology, doesn't demand any lab, wet lab. You just need to have some understanding of computers. So before I proceed, I also want to know how many of you are familiar with Linux? That's very important. Unix and Linux. I'm sure most of you are aware of Windows, but how many of you are aware of Linux? If you can give a shout, yes, you are aware, then it helps. If you are not, then I will proceed accordingly. Okay, looks like none of you are aware about Linux. So, which means that I'll have
Basics, sir.
basics means what?
Means like Linux system, like MacBook handling and this kind of thing. And like opening up directories, changing directories.
So that so mostly the interface of Linux is now almost equivalent to that of Windows, right?
Yes.
So so anybody who uses Windows or Linux can interchangeably use it because the desktops are very identical. So besides your ability to use the desktop, are you aware how to use the command prompt, how to execute shell scripts, how to write a few lines of code? Okay.
No.
Yes, anybody who knows it? Okay, so that's fine. That's not an issue. So because I know that you don't know, then I will go slow and I will try to, not cover all what I have to share with you. And I will try to make sure that whatever part I teach, uh, it's understandable. And in case you have any question, feel free to prompt and and we can go the question asked. Okay, so first is the basic. So what is a database? Okay, so I'm sure everybody has heard this word. So database is nothing but a collection of structure structured, searchable, periodically updated, and cross-reference data. Okay, so this is basically a database. Okay, so now for those of you who do not know programming or are not aware of uh, computers, I'm sure all of you have used your dictionary, Bengali dictionary or say English dictionary. So in the dictionary, dictionary is also a database, right? It's a database of words, and the words for each word there, you have some explanation, right? And then you have also a pronunciation, right? And then you also have synonyms and antonyms, right? So for each word in the dictionary, so you would have several fields, right? So you have an English word, and then you will have its pronunciation. You will have, uh, mm, a text which explains it, and then you will have synonyms and you will have antonyms. Okay. So the organization of this dictionary is structured, which means that there is an order in each line, which means the first the word that you're going to search for is there, and then the pronunciation is there. Thereafter, you will get the explanation, followed by the synonyms and the antonyms. Okay. Now, you have to search this database, right? This dictionary. So typically when you want to, search a word, what do? You don't go and, just start from page 1, right? Because you know that if you do that, it will take you long time to reach to the word of your interest that may lie somewhere in the middle. If you are very lucky and the word is in the front page or in page 1 or 2, you're very lucky, right? So typically what we do is we go to the back, and the back pages, the glossary has a mapping, right? And there you will have the word, and the word is mapped to the pages, right? So if you go and check the word there, the words are sorted there and they are indexed, which means there is a location information which points back to the position where it exists in the main dictionary. So that is what is an index. Okay. Sometimes some dictionaries, also allow you, to search by the first letters or first two letters. Okay. So you will see some dictionaries have the letter L or LL, carved out of the pages so that you can quickly go to that page and you can find it out. So this is also some kind of indexing. It's called bucket indexing. So these kinds of organization or structuring helps you to find the information very quickly. Okay. Now, given that these information are all present in the computer, obviously you also need associated tools to get the information quickly. So these are called database access tools. And then you need to update the data regularly, and so you would need to tool for deletion, some corrections may be necessary, and so forth, right? So in whole, you need what is called a database management system. Okay. So the database management system will handle all the data and will allow you to interact with this backend and will provide you the information very quickly. Now the question is that why do you need all these things? Okay. Now for a small, notebook that you have, on your desk, this may not be necessary, right? Because you can as well, go through the pages and quickly find it out. But imagine, you know, the data that comes out of the human genome. Okay, the human genome itself is 9 billion bases. Okay, and correspondingly, so much data every day people are generating. If you have to actually, browse that information or search for anything, it is impossible humanly to do any of these tasks. Okay, so we need databases and we need search tools that are efficient. Okay, so that's what is a database.
Sorry, sir. Extremely sorry for interrupting. So
Yeah.
there are two respected participants raising their hands. I think they have some queries.
Yes, I'm not able to see where are they raising their hand.
Uh, so Mr. MD Iqbal Kurasi sir raised his hand.
Yeah,
I think
please.
he has some
Yeah.
queries.
Yeah, you can ask a question.
Sir, actually, on that time when you asked those who know Linux, just say, so we just put our raise our hand. So without interrupting you, just telling you that we know Linux. That's all, sir.
Okay. So is there anybody else? How many others know?
Sir, it's fine, sir. There are no one, sir.
Okay, so you are the only one who knows. So you will have to bear with a little bit of low level information so that others can catch up. Fine?
Yes.
Okay. So this is a simple example. Okay, so I have 3 entries. These entries are on in flat file. Okay, flat file means the simple text files that you can as well write. Okay, and you can also keep these data in what is called tables. Okay, so when you keep the data in a table, then it is more manageable. Okay, manageable to whom? Manageable by the uh, computer, because then it is very easy to relate to each of the information that one needs to access, because each parts of the information are broken down and kept separately. Okay, here you can see there is one record and these are given as name, last name, accession number. Okay, and you can see it is broken down here. So under teacher name it is given here, under accession number the information is given here. So they are all separated out and you can pick and choose whatever you want, right? So that is how it goes. So this is how we do the searching. Okay, so we need biological databases, as I said, because they are extremely big in size. There is a lot of data that are accumulating in petabytes, and we cannot deal with the data otherwise. So what kind of data we have in the biological databases? So mostly I will concentrate today on giving you overviews on molecular biology databases. So these would consist of one-dimensional data like sequences, okay, and then you have three-dimensional data like structures. You have image data like 2D gels, okay, and you have other kinds of data like text data. So there are various kinds of data, and the databases are also organized into various categories. Okay, so before we actually look into some of those, uh, let me also tell you that this area itself, you know, is so big that even you even have journals that only publish databases. Okay, which means that researchers are doing research and publishing databases. So what these journals do, they look if you have gathered some information from primary databases. So there are different kinds of databases. Okay, uh, there are different kinds of databases. The first is called the primary database. This kind of database sources its information directly from the experiments. Okay, it may not be the raw data directly, but it may be very close to the experiment. For example, sequence. Okay, sequence of the DNA is a primary data. Sequence of a protein is a primary data. Okay, but say if you take the sequence and you do some calculation, see you find out what are the hydrophobicity distributions of all the amino acids, and use that data. You chop out all the parts of the sequences and you organize it to some kind of a new information format, then it becomes a secondary database. And if you have found out some interesting information from that data, then you can publish this in various journals. So there are some journals which only publish database, and there are some other journals which bring out special issues where you can publish these databases. Okay. So before we go any further, I actually want you to have a peek actually what goes at the back end. Okay. So that you know, sometimes we are only looking at the front end, but if we look at the back end, then we also have an understanding what goes on behind. Okay. So we will quickly stop sharing and then go to let's see. Okay. So are you able to see the screen here? Okay.
Yes.
So what I'm going to show you is a back end and a front end, so that you have a fair bit of judgment what's going on, and this will also help you understand how people build databases and what the information that you're getting, how it is sourced and organized. Okay. So let me first show you one of my databases that I have created. Okay. So this is one database that we have created in our lab. Okay. So as you can see, the website is public. It is viewable all over the world. And this is a secondary database. So what it does is basically it takes the genome sequence, and from the genome sequence, it identifies the intergenic region between two genes. Okay. And it identifies the intergenic region from an information already present in a file that we download from the GenBank database. Okay. And GenBank database, as you know, is provider of primary data, which is actually genome data and gene data. So we identify the intergenic region, and in this intergenic region, we identify transcription terminator sites, intrinsic transcription terminator sites. And then we provide these information back to others all over the world. And then people use this database heavily. It's one of the very highly used databases. So you can see an overview is given. And for those who are interested in knowing more detail, you can look at the paper. So you can see that there's a front end, there's a dashboard where you can enter some parameters. Okay. And then if you want to search, then you would have to enter parameters and then you have to submit. It will give you the results. And there are various kinds of possibilities. You can search for archaeal genome, you can search for bacterial plasmid, archaeal plasmid, and so on and so forth. By default, it does the bacterial genome search. Okay. So, and if you want to not search but you want to browse, then you can go by these letters. So if you click, then you will get the name of the species, and then you can click on the species of interest. For example, if you're interested in K12, E. coli K12, we can find the information which is given here. Then you can browse all the information. And if you want to know for every gene, then you can go to the genome viewer and you can find it out. So I think this is gone a bit outside. Okay, so each of these are terminators, and you can find them out by simply clicking, or you can choose a gene of your choice. Okay, and you can go to that window and you can find the terminator for yourself. Okay, so this is a simple example of what a database looks like. Now, how do we do this? So to do this, you need to know a little bit of programming, and then these kinds of stuff are essentially done by students in the lab. Okay, so the research students do this in the lab and generate these kinds of websites. So I will show you how it is done by quickly giving you a broad idea how the information is organized and how it is accessed. Okay. So first, let us look at the information. Okay. So you can see here uh, there is a prompt which is called MySQL. Okay. So MySQL is basically a database, okay, relational database, like what you have, like Oracle or Postgres SQL. Okay, so these are names of different kinds of relational databases. Okay, and you can uh, mm, create a database. You can store the information here, and then you can access this information for your research work, and you can also disseminate this information through a website. Okay, so you can see that in my server, the server is a computer which serves the commands that are given by the user or the client. Okay, so in this case, you can see my server has many databases. Okay, so among one database is the web gesture. Is my cursor visible? Is my cursor visible?
Yes, sir.
Okay.
It's visible.
Yeah. So among others, you can see that different databases, we have one database, it's called web gesture. So this is the database that we quickly had a glance a while back. Okay, so if we want to look at what is there in this database, so we have to say okay, we're already using it, so there is no so it is itself a database. So what we have to say is show tables. Okay, so we use the uh, database. So when we say use database, so use web gesture, so it picks the web gesture for its activities, and within that database all the information is uh, organized in tables. Okay, so you can see there are various kinds of tables here. One table is for bacteria, one table is for archaea, then there are keywords, and then organism names which are from plasmid, and then there are taxonomic information. So all the information is input here. So if you want to look at the tables, say I want to look at the tables of bacteria, how it is organized. So you can see I'll quickly increase the size. Okay, for some reason the window is very small when I load it in Zoom. So you can see that all my information is organized in some manner here, and it is loaded. Okay. And I have some kinds of, names for each of these fields. And then I declare, whether integers or there are, decimals or there are texts. Okay. And then I store all this information. So for all the tables, I will have data like this. Okay. So you can see for taxa also I have data. And each of these data tables are fully loaded. Okay. So if I want to see how many lines are there in these tables, so you can say select star from say taxa. If you just say like this, okay, then it will show you all the information in the table. Okay. But if you want to look at the number of records, so you have to say select count in bracket star from taxa. It will tell you number of records. So this is how you get all the information, and then you store them in separate tables. So you can see this table has some 257,276 entries. Okay. And this covers the total number of species that we cover in our database. So now taxonomy of species, not species, taxonomy of species that we cover in the database. So now each of these tables will also have some common, data using which you can link between data, two tables, and you can extract information. Okay. So these are called common keys, and this helps you to join the tables and extract information. So these things are known by persons who know a little bit of programming, and these instructions are called structured query language. Okay. So this is all for information, and I'm showing you the real thing so that it gives you some flavor of how these things are done. And remember that nobody is going to do these things manually, right? Once you will give your input from the website, okay, there is a program. This program is called API, okay, Application Programming Interface. That program will convert your question or the query into this structured query language and extract the information from the database and return it to you. Okay. So this was the database that is as is stored in the backend. And remember that whichever database you will see from now on, in the backend, the data is stored exactly like this as I have shown you. Okay. Now let us look at the front end. Okay. So I will quit this. I have already quit this database. Now I will show you how the front end is, done. The front end in my case, we will go back quickly uh, to the web page. Okay, so you can see that the front end, has the address, and then it has a directory under which, some files are there. Okay, and these files are called hypertext files, which gives links to other pages, and these pages again, uh, are active pages which has code that takes the information from you and returns the answer. So for example, in this case, you can see that there is a code here which is written in a language called PHP, which takes the input and returns the result. Okay, so what we need to do is we need to go to that directory. So I will change over. Okay, so you can see whatever you saw on the screen is stored as a directory. Okay, and here the directory gesture is there, and in this that file is there, db search.php. So this file is there. So this file has all the information. Okay, so this is a small file which just takes the input. Okay, it just takes the input and then it sends out the queries. So now you may want to ask, how to do this. So you can see that this file itself, has instructions embedded here. How to convert the query from from yourself, and then put it as a variable into the code, and then interface with the database, extract the result, and display it back to you. Okay, so there is the main function, the main program, and there will be small subroutines that this program will also call, which will do small jobs. So in total, together will do all the jobs. So I don't want to bore you with any of these things. I just want to show you the backend so that you understand that what you see and what you have at the backend, how they gel together. Okay, so sometimes we don't know, we wonder how these things are done. Okay, this was just for your uh, information. Okay, now the question is that am I only able to do these things using PHP? No, not at all. PHP is just one of the languages that one uses. You can use other languages like uh, CGI Perl, you can use Java, there are many others that have come out, recently that you can use. Okay, so there is no hard and fast that you have to use what I have used, but each of them have different syntax, and you would need to know the syntax to implement these concepts. Okay, so let's go back to the original PowerPoint. Okay. So now, if we look at the current status of biological databases, you know, we have more than 5,000 different databases, you know, that one can actively search and get the data from. In fact, in many labs, small interns, you know, in their 1 or 2 months, they do these kinds of small databases to, you know, to get a hang how to retrieve information and how to archive information and how to quickly process information for useful knowledge. Okay. So generally, to get access to how to find out, first of all, you know, uh, which database stores what. Okay. Given that there are 5,000, more than 5,000 databases all around the world, there is no, you know, list that will tell you that, okay, these are the URLs which this kind of data, these are the URLs with this kind of data. No, it is not. So you'd have to do a Google search, or if you have read a paper, the paper may have given you some URLs that you may need to search. Okay. There are some universities and some, you know, groups who may, you know, uh, who may make a list, and that may help you. Uh, is the um, is the browser visible as well, aside from the slides?
No,
Hello?
no.
Okay. Is it now both the slide and the browser are visible?
Just the browser is there. Slides are
Browser
not.
is visible, sir. Slides is there.
Slide
Sir,
is-
parallelly you can visit it. Yes, sir. Yes, sir. Yes, sir. Partially it is
Okay, great. Yeah. So for example, I have clicked on the bioinformatics.org website, and you can see that that this website, you know, also allows you to browse alphabetically. And then if you go to some, you know, if you want to browse, you can see pancreatic cancer genes database. Okay, so let us click. So you can see I have clicked it, and it has taken me to some, you know, some site for which this is the home page. Okay, so this has taken me to a pancreatic cancer gene database. Okay, so I am coming to this database for the first time. I did not know that this database existed. So I simply took help of another website, which, I mean, it's a voluntary. When people do their databases, they can come and register themselves. So this group has registered themselves and have given a link. So now I can go and browse this database. Okay, but it is at your own risk, because nobody guarantees uh, what information is there unless there is a peer-reviewed paper, okay, that has looked through the database and has approved it. Okay. Otherwise, you can also do what I suggested. Is you can do a simple Google search. Okay. So what is the current size of data? So we have lots of data. The size has exceeded 1000 TB. The protein sequence data is also very large. It's more than terabytes. The 3D structure data is also very large. And the updates are made daily, bi-weekly, monthly, annually. So it depends, from database to database. Okay. And there are many databases, and it is impossible for one person, as I said, is more than 1000, 5000. So this is just, I'm giving you a snapshot, some database names. Okay. So ABCD just goes on and on. Okay. So there's some history of how these databases have evolved. So in the very early days, when computers were not so available, so the data was mostly available from books and articles. Okay, and then once the computers became a little bit powerful with the advent of the desktops, then much of the data used to come in form of tapes. Okay, uh, and then floppy disks, but tapes were much you know um, could store much more data than floppy disks. And then by the 90s, CD-ROM came, and after that, the internet, the FTP servers started appearing, and the online services, slowly they crept in. But by 1993, when the internet started taking hold, we were able to download data directly from the URLs without use of FTP clients as well. And as the data increased, CD-ROMs went out of fashion, and DVDs have come. So nowadays, so if you want to buy information outside your regular downloads, these will typically come in flash drives, pen drives, or DVDs. Okay. So the categories of databases I've already exposed you to. So primary data, this is data that comes directly from the experiment or very close to the experiment. Maybe it's the raw data and then it has been just processed to eliminate unnecessary information and being given to you. And then you have secondary data derived from the primary data. And then you would have other kinds of databases which you can compute or you can extract from information such as the bibliographic databases and so on. So, okay. File format. Okay, so this is one more important thing to know, right? So we already looked at tables. Okay, so the tables that we looked at in the computer were MySQL tables. So MySQL has another version. It's called MariaDB. So most of the relational database tables come in the format of the software, right? Which means if it's Oracle, Oracle will have its own way of formatting the tables, but mostly it is a column and a row. It's a table, so it's always a column and a row. but there would be some minor differences in the data types and what is acceptable and what is not. When it comes to flat file, which means files that are human readable, you have many different file formats. Okay, so a quick example is the FASTA. I'm sure all of you know FASTA. FASTA is the file for storing sequences. Okay, but in the older days, it used to come in GCG format, the PIL format, the MSF format. Okay, and still, as far as I know, there are some 16 different formats for just storing sequence data. Okay, so there are no standardized formats because of the simplicity of the FASTA format. It is usually the one that is most popular, but it's not necessarily so that everybody uses it. So the question is that if I want to create a database and I have to access data from different types of file formats, then there is a tool, it's called Open Babel. So this tool is available free of cost and you can download it. So as far as I know, it probably also has a Windows version, and you can install it and you can convert uh, files among different formats. It is very popular among students. So when you have to build a database or you want to access a database, so the idea is to store information that everybody else is going to find useful, or you have to want to store some features that is important for doing some kind of research. Okay, so that is how the structure of every database is designed. So minimal content, nevertheless, is what is shown here. So obviously you need to have the sequence. Then you need to have what is called an accession number. Okay, which means a unique number that always maps to that record. It must also have some references, which means that who deposited the data, who generated the data, whether the data is peer reviewed, it's curated or not. So those kinds of historical information is required. Then you need the taxonomic data, which means which kind of organism. And then you need annotation. Annotation is essentially a analysis of the sequence, right? This is a secondary information that you generate from the primary data. Then you want have keywords so that others who do not know about the sequence want to find a sequence based on some keywords. And then you want to have cross references. What are cross references? Which means that this kind of information may be also present in some other databases, right? Yours is not the only database. Maybe you don't want to give all the data. You have extracted data, part of the data, but you still want to tell the others that, hey, if you want to get more information, you can look at the other database. So you can use the cross reference. Then there's a documentation, telling, about the sequence and anything that you want to put. This is essentially a free space for you. This is one example of biological flat file. I am showing you the flat file. The reason is that if I have to show you the HTML page, then I may not be able to show you the all the information that is actually there. So typically what happens is that the files are there, and then your HTML browser reads this file and then puts it in a certain format so that this is visually pleasing to you. Okay, but at the back end, the information is stored in a flat file format. So this is an example from the Uniprot Swissprot flat file. So by the way, Uniprot is a repository. So it is for a universal protein sequence database or universal protein database. That is why it's called Uniprot. Okay, so you can go to the website www.uniprot.org to get all the data. So this Uniprot database is essentially an amalgamation of several databases put together. So you can see that is the same protein here. So it's a sequence data. You have the same protein here. This record was entered and updated last time in 2001. And the same data is now updated again on 7th April 2021. Okay, so you can see that the base data remains, but it is being updated with more and more information time to time. Okay, and every line has an indicator here. So it tells what this line contains. Okay, so for example, CC means comments. Okay, DR means database reference. KW means keyword. R stands for reference, and RP and all these stands for different things. Okay, OS organism source, DE description, DT date, AC is accession, ID is ID. So all this flat file is organized in a structured manner. Okay, so that it is easy for you to get the information. And most websites, will change format, but underlying content is same or updated. Okay, so if you want to know what the information, sorry. So if you want to know what the information is, and you want to actually use this information, so typically what we do is we download the database. Okay, now you will ask the question that shall I go to every page and download one page at a time and save it and then read it up? No, absolutely not. Okay, so there are several ways to do it. One way to download database is to use a command in Linux. It's called wget. Okay, so for those of you who want to actually get started with um, downloading databases or web pages, must look at the command wget in Linux. Okay, and for those who are already into some programming, for example, programming in say Perl or Python, in each of these there are download agents. Okay, the download agents are nothing but small libraries which you can program, where you can pass the URL information and then you can download the information from the database one at a time. Okay, programmatically. Some websites, some databases, I will show you, allows you to download the whole database in one go in a compressed format, but some don't. So in those cases, it is manually impossible to download one at a time. So programmatically, you can do them. Okay. So if you're interested to look at uh, the files, the UniProt files that are provided, you should actually go and look at so this is the page. I've already kept it open. You can go and look at this page, and it will tell you, what these lines are. So you can figure it out. Each UniProt database page is very rich in information. Okay. For example, if I go to UniProt now, okay. So this is the UniProt database. Okay. And our protein was erythropoietin. Erythropoietin. Okay. So it has returned us the results. Okay. So you can see that it returned us the result, and we were looking at the human, the sequence from the human. So this is the entry. So all the information that I'm showing you in the flat file, okay, this is nicely organized here and is given. Okay. So what it is doing, it is taking your sequence information, it is parsing this flat file, then it is going to the cross references, going to those databases as well, gathering all the information, and together it is showing you this web page. Okay, you can see it is not just the sequence or the taxonomy. There is much more information in this. So how is it getting it? It is not only getting the information from this page alone, but all the other cross-reference databases. So that is what is given here. So this single page gives you almost all the information that is cross-referenced by UniProt. Okay. So you can see this is a huge list of information that you can get, and it is nicely displayed. Okay. Okay. So now we can look at the nucleotide sequence database. So if you're interested in nucleotide sequence database, similarly, you can go to the NCBI GenBank website. So this is the URL there. And if you give a query, again, we gave a query for that same, erythropoietin gene. And you can see this is an old entry, and this is a new entry. Actually, it's not an old entry. It's a full entry that it gives out. So it will give out the exons, the gaps, the whole gene, the source, the references, and everything. Okay. And remember, when you went to the UniProt, you actually had all this information in one shot. Okay. And these are all hyperlinked. So if you want nucleotide information, if you click it, it'll actually take you and get this information in another page. Fine. So that was nucleotide database. Then we look at, say, genome database. So in genome database, typically we are looking for genes, gene location, nomenclature of genes, links to other sequence databases, organisms. Okay. And then you have organism specific genome databases also. For example, the Mendelian inheritance in man, the genome database, the mouse database, the Drosophila database, yeast database. Actually, there are many, many databases. As I said, it's impossible for us to cover all of them. You can go and check it out yourself, those databases that you find interesting. In all cases, the formats are generally, you know, relational. Typically, they are Oracle or MariaDB kind of database. Okay. So this is just a snapshot of the OMIM database. So it is essentially a catalog of human genes for monogenic disorders, and it gives you description from abstracted from literature and picture and their original references of all cataloged human diseases, monogenic diseases. Okay. This is one snapshot from the database. I mean, if you are interested in certain proteins, you can look at it. You can see it has text that gives you references and then where it occurs in some creation date and things like that, contents. Okay. For the most popular, currently most popular sequence and genomic database, if I have to say, then these are the two resources you should be looking at. One is the Ensemble database, and another is the SRA database. So Ensemble database is essentially providing you all genome information, including the gene and related information. SRA database is giving you experimental data corresponding to the genomes. So high throughput experimental data. So as I said, Ensemble contains all genome DNA sequences, annotations about genes, polymorphisms, repeats, and then homologies. This is maintained by the EBI, the European Mathematics Institute in UK at Kingston, and the Sanger Institute, again in UK. You can search for the DNA, you can browse for the chromosome, you can find matches, closely matches with the mouse, look for protein families, give a threshold and get matches and things like that. Okay, just one snapshot of Ensemble. Okay, you can see, these are all hyperlinked, first of all. Okay, which means that this image that I'm showing you is actually, if you put your cursor any of these places, okay, these are called interactive maps. So if you put the cursor here, then it will show you that this place is active, and if you click that place, then it will give you more information. Okay, so that way you can zoom in and get more resolved information. So typically, you will get the coding genes, the non-coding genes, long non-coding genes, pseudogenes, repeats, variants, and so on and so forth. Okay. SRA database, the sequence read archiver. Okay, so this is a database which has become popular ever since high throughput sequencing, came into vogue, and lots of genomic data is being generated. So if you want to do your own analysis, so you have to go to this database. Now the fact is that you can, this database gives you FASTQ files which are essentially the readouts from the experiments. You can see these databases are very large. So, number of bases in this for this set is 10.4 gigabases, and the size of this is 4.3 gigabytes. So typically, you need 20, 30, or 40 gigabytes of disk space at least to download the data, and the data is downloaded in a compressed format, and it is not downloadable directly. It is downloaded. You have to download a toolkit, and through this toolkit, you would be able to get access to the data. So that was a snapshot about nucleotide sequences and genome sequences. Uh, Next, we go to protein sequences. So as we saw, all protein sequences are actually stored in UniProt these days. A few years back, about 10 years back, uh, the sequences were actually mostly, downloaded from Swissprot, TREMBL, or PIR, or the RefSeq databases. But currently, uh, we have two major sequence archives. One is hosted at the NCBI website, at Bethesda at USA. Here you can get sequences to the RefSeq database, or you get your sequences to the UniProt database. So UniProt database is essentially a collection of information of the Swissprot, TREMBL, and the PIR PST database. Okay, the information from these databases have been merged in through a consortia project. It's called UniProt. We have already looked at a UniProt web page through a search, so I'm not going back showing it to you again. There are other protein sequence databases such as PRF, GenPep, and there are many other specialized databases for specific sequence families or group of proteins. We will look at some of these as we move on to the next slides. So UniProt, I've already given you a brief. So good part of UniProt is that they're mirrored all over the world, so it is very fast, and you can access it programmatically, or also you can access it manually. So this is overview of what is there in UniProt. Okay, so UniProt comes as UniProt knowledge base. So you can see that so many databases. As I said, these are cross-references. So these cross-reference databases, are used to gather data, and also sequence analysis, manual curation, and then whatever predicted data on classification are also obtained, and these are pre-calculated and stored. And when you give a query, then these are accessed and are given to you. Okay, so this very comprehensive information at your fingertip, delivered very efficiently. As of January this year, this database had some 208 million. I think, yeah, it's a 208 million entries. Okay, and these were derived from the Swissprot and the TRML entries. And there are several sub databases, which means that out of 208 million entries, okay, um, some 262. I mean, this is a non-redundant entry. Okay, uh, when we take common entries, redundant entries, the total entries are 262 million. If we apply a 90% identity criteria, there are some 130 million. And if we apply a 50% identity criteria, some 49 million entries are there. And this UniPark is essentially a cross-referencing database, which means that you can uh, take the UniProt ID, the SwissProt ID, or the TRML ID for a given sequence, and using this UniPark, you can get the unique ID for other databases. Okay, so this UniPark is very useful for bioinformaticians who want to create new databases with new useful information. So when it comes to other databases, I think I've already gone through this. Yeah. So when it comes to other databases, PIR, PhD is there. I think we have already gone through this. So PIR, PhD currently, hosts several new information. For example, post-translational modification, or networked information that is accessed through literature search. You can also do a peptide-based search of amino acids. So different kinds of toolkits are given to you to gather information. Okay, this is just a snippet of information from the PIR PhD data set.
Professor Paul,
Yeah.
may I have a request? If
Yeah.
you can just open, if you can just show them the, as you are talking about the protein database, so PDB, okay, and this is the rcsb.org from where they can download this PDB things.
Yes, yes, I will once I come.
And actually, if someone opens a PDB file, so in the WordPad, what the informations are there, they
Yes.
can get.
Yeah, yeah.
Okay.
I have
And
slides.
also one more thing is to use XPacy.
XPacy is actually now very limited because most of it is now available directly through UniProt.
Yeah.
Okay.
But XPacy, although it has a now limited, but actually why I'm saying is in some of the courses, okay, they have the XPacy is in their syllabus. Okay. So if you can.
Yeah, so for those of you who have XPacy in their syllabus, what has happened is that earlier XPacy was the sole source of sequence information for both SwissProt and TREMBL. Okay, so SwissProt is essentially a service given by the Swiss Bioinformatics Institute, and TREMBL was a service that was being given by the European Molecular Biology Lab. Okay, now what has happened is that since the sequence archive became bigger and bigger and bigger, it became very difficult for all of them to search so much information at one go. So that is why three sequence repositories, the Swiss Bioinformatics Institute, the European Bioinformatics Institute, basically the EMBL, the European Molecular Biology Labs, and the Georgetown University, which hosts the PIR PhD, they formed a consortia, and they amalgamated all the information together, and they are essentially giving all the information, non-redundant information, that existed at that time in these three websites together in UniProt. Okay, so for that reason, some of the things that you originally had from SwissProt will
Oh,
immediately come in UniProt.
why I am saying is, if you have suppose a genome sequence,
Yes.
or if you have a protein sequence, so
Mm.
what is the root? If the translation after that, so expressive from that you can get it. Okay.
Yes. So what I'm saying is that you need not have to go to different databases. Through UniProt, you can walk the page and you can get access to that information.
And we have only half an hour more. Okay.
Yes.
Okay.
I try to cover
Yeah.
some of the databases. Since there's a lot of information, I will try to just give a brief, and for PDB especially, I will give a little bit more detail.
Yeah.
Right?
Yeah. So okay, but in case of UniProt, if some conversion of the proteomics, you can tell it also tomorrow also, because you have the you have to
Sure,
take
sure.
continue. Okay.
So once you have the protein sequences, as we saw here in this PIR-PSD, they're giving you post translational modification, or whether any protein is a substrate of any kinase or not. So all kinds of other information, useful information, are being gathered and are being kept. Okay, so these information are unique to these repositories, because the common things they are actually now only giving out through the UniProt. Okay, so this is one example. So from sequence, you can find the protein domains or protein families. So there are databases for that also. So some are based on patterns, some are based on Markov models. So protein domain, or ProDom, is one such um, you know program. So as you can see, there are several algorithms that are used to make these. And once you have the information, you can get some interesting statistics. There are some examples. Just go over. So this is ProSite, is another secondary database that will give you patterns, small sequence patterns. PFAM is give you domain, but ProDom also gives you domain. ProDom gives you structural domain. PFAM gives you functional domain. So sequences are analyzed, and then these are uh, kept in different databases. Okay. So instead of actually finding the information individually, okay, so which is kind of tedious. So what has been done is that a composite database has been formed. It's called Interpro. Okay. So instead of going and finding all of this individually in one shot, if you do a Interpro search, okay, you can get this derived information. Just like in UniProt, in one shot, instead of going to SwissProt and trying to find something or somewhere else. So these are the main databases you should try to search to reduce your time of getting results. Okay. So ProSite, as of now, it has some 1883 documentations, some 1311 patterns, and so on and so forth. So each of these you can get, and they are well annotated. These are some of the information. So just like in UniProt, you can see that every line has, has a key, which tells you what the information is. You can see when the database reference, which means in which protein in UniProt you can get this match, and that way you can interoperate between sequence and patterns. So this is one entry, a documentation entry. So for each pattern you have a document, you can browse it. So you can also browse the all the alignments, all the profiles from which the pattern has been derived. So these are very useful for bioinformatic analysis. Okay. So ProSite also these days try to annotate function through domain assignments. So these are called Pro rules. So there is a page for that as well. PFAM, as I said, PFAM refers to protein families, but these protein families are function families. And the families are generated by sequence alignment through hidden Markov models. And this is one example of the same protein, erythropoietin. So you can see the domain here. The functional domain is from residue 31 to 191. And then from other programs, it suggests that 1 to 27 may be a signaling sequence. Then 7 to 32 is a low complexity region, and 145 to 156 is low complexity. A low complexity region means that here there may be repeats, and because there are repeats, the sequences are not very unique. So maybe only a few amino acids are continuously being repeated. ProDOM, again, this is structure based, and alignments made from domains clustered from the SCOP database, release 135. So SCOP, you don't know, it's a structured database. We'll look at it in next few slides. So you do basically alignments against domains that are identified by structures. Okay. This is one example after alignment. Okay. So print is a motif fingerprint. So I'm essentially telling you what information you will get from each of these individual databases. So as this name suggests, fingerprint gives you a set of motifs or fingerprints for a compendium of proteins. So this is the InterPro finally. So currently this InterPro has so many entries. Okay, so you can see that we had so many sequences, right? But after grouping them together, okay, they have been grouped into only 38,633 entries, which means essentially so many families, you can say, in some sense. Okay, so in this you have homologous superfamilies, family information, domain information, repeat information, and site information. So in one shot, if you give a sequence for search in InterPro, and it will find the family, and within the family it will give all this information to you. So you need not have to do individual searches. This is one snapshot. You can see ProSite, PFAM, the publications are there, sequence IDs are there, some abstracts there. So this kind of information you will get. So there are other databases, mutation databases, like human gene mutation database, variant database, genome-wide association database, DB-SNP. There are disease-specific polymorphism databases also. These are some of the names: SAP, CSNP, and so on. DBSNP interface, this is the most popular resource. It has some 328 million submissions till date. And if you want to identify SNP for a given gene, then it gives you very rich information. These are marked here. So if you're interested, you can go and have a look at it. SNP databases are generally modest in size because they are only archiving the altering information. They don't archive the full information, and there are many formats. So there are currently initiatives to harmonize them. Then we are coming to proteomics. Okay, so the last slides we had only covered sequences, as we said. So first we covered gene sequences, nucleotide sequences. Then we covered genome sequences. We did not cover RNA sequences. Okay, so but there are RNA transcript databases also. Okay, that you can these transcripts you can download from the SAR website. Then we looked at the proteins, then sequences, and then derived information such as domains and fingerprints and motifs and so on and so forth, and mutations. When we come to proteomics, we have 2D images, and these are some of the databases which store these images. Okay, Swissprot can be used as a master index. It can give links to these. There are not many protein mass spectrometry databases archiving data sets. PRIDE is one of them. So this is one image that you can download from Swiss 2D page. This is a front page of the PRIDE website. So if you want to download maybe cancer data, or if you know some ID, then you can give the IDs here, or you can search by text and you can download the corresponding mass spectral data from this website. Coming to 3D structure. So the main, the spatial coordinates are typically from X-ray or NMR, and recently the cryo-electron microscopy data sets are also increasing in number. And so the primary data comes from the protein data bank, the PDB, and then the secondary data consisting of the classifications come from the SCOP database, yes, CATH database, and then you also have databases like BMRB, which houses the spectral information from NMR. Similarly, you also have a database which houses the electron the electron map data. Okay, so there's the database for that also. Okay. Then you have fold classification database, homology derived secondary structure database, and so on and so forth. So the PDB is basically managed by a website called RCSB. So if you say rcsb.org, then you will get the protein data bank. It has proteins, protein nucleic acids, virus capsids, and so on and so forth. It allows visualization, and the website has improved a lot, and it is now also information cross-reference from many other databases. So currently there are about 145,000 structures and about roughly about 90,000 unique molecules. Okay. So this is an example of the PDB data. So this is what you will get if you open a protein data bank file. So as you can see, just like in the sequence data, you had 2-letter codes, okay, like AT, AC, ID. Here you have these, 6-letter, keywords. So there's a header, there's a compound, there's a source, author, data validation, the journal where it's published, and then there are remarks. Okay, and within this remarks is again divided into categories. Okay, so for example, remark 2 will tell about the resolution. Remark 3 will tell you about the refinement parameters in the crystallography. Remark 4 will tell you some other information about termini, mutations, and so on and so forth. Okay. So you also have secondary structure information directly written onto the PDB file. So you will get secondary structures like turn, sheet, and you can see that the residues and where exactly these are present. Okay, these numbers are positions that are given. The turns. So to read this file properly, the PDB website also gives you a manual where all the formatting information is clearly described. So you can find out each of these cases, what it means. And the format is given column wise, which means that although you are seeing that there is some kind of jitter here, actually this information is perfectly aligned in the text format. Okay, so the main information actually starts here, the coordinates, and here the line numbers for the atoms are given in order, and these are the atom names, these are the amino acid residues, these are the residue position in the polymer chain. These are the 3 X, Y, Z coordinates. This is the occupancy, and this is the temperature factor. So for those of you who do not know about occupancy and temperature factor, Professor Banerjee in the class, I think in the morning, may have given you some introduction. Otherwise, you can refer to some crystallography manual to see how these are calculated. Okay. So we can also quickly look at the website, the RCSB website. So the web browser is visible. Hello?
No, no,
Yes.
no.
Okay, just a minute. Yeah. Okay, so you can now see the website here, right? So, if you say if you want to find a protein for which you don't know the code, you can say, for example, I want to find, say, hemoglobin. If I write hemoglobin, it's going to go to the UniProt, and it's going to find out, what the different ways it is written. So you can click one of them, okay, and then it will give you all the corresponding structures, and you can take it as a different kind of a report. You can reformat it. You can refine your search say, if you want to only find the latest ones, you want to do some refinement, you can click these check boxes and you can refine your search. Okay. There is also an advanced search. Okay. There's also an advanced search where you can make a complex query and you can give your search, or you can do a sequence-based search. You can make a motif-based search, and you can also find all the ligands, chemical formula, which is present in some PDF file. So you can do different kinds of searches. Okay. So remember that exactly how initially I've shown you the whole thing works. Your query is taken and converted into some kind of a query language, and then all the information is given back to you in a nice format. Okay. Okay, so I think we are almost at the end. I have few more slides and try to cover those. Okay, so the other databases are like pathway databases, metabolic databases. So some popular databases are like enzyme database or the Brenda database. Okay, so if you go to these databases and you give, say the EC number, so you can search by EC number, you can search by gene name. Okay, anything that about that enzyme, so it will give you all the information about the chemical reaction, in which species it is found, and what are the products, and so on and so forth. Okay, there are specialized databases for organisms like E. coli, and then curated database like the KEG. KEG is the Kyoto Encyclopedia. Okay, so these databases, have very, you know, very rich, maps, clickable maps, because they are showing you pathways. So once you click, say, in a KEG map, so you can look at sub pathways, and then you can go to sub pathway. You can click on a gene, they show information of gene. So essentially, you can zoom in and zoom out, and then you can gather the information that you are interested in. Then there are bibliographic databases. The most popular database currently that people are using is a Google Scholar. Okay, so Google Scholar has archived information from various databases and gives you information on patents and papers. It'll give you the citations also, which papers have cited your paper, and give you the abstract and other details. There are also specialized databases, for example, agriculture-related databases called Agricola. Many other databases like that is there. If you're interested in a particular domain, you have to go and find them out. PubMed, if you are not using Google Scholar, you can use PubMed. So PubMed essentially, has assimilated all the data from old Medline, Hellstar, etc. And after that, it is archiving all the new bibliographic information integrated with information from molecular biology databases. Okay. So you can refer to it through PubMed ID or Medline ID. Okay. So other databases that it connects to are some like Rebase, Transfac, Medops, Pronet. There some have actually become obsolete as well. Okay, and these keeps changing from time to time. There are some databases which cannot be actually classified into anything related to you know, sequence or protein or structure. So just like KEGG, you know, it's a pathway database. This is a string. This is a functional enrichment analysis database, a protein-protein network database. So it has some 24 million proteins, some greater than 200,000 interactions, million interactions, and some 5,000 organisms. Okay. So I'll come almost to the end of my talk. So people ask, what is the best database for sequence analysis? So my answer would be the UniProt database. So if you want to analyze data for sequence, UniProt will give you the most comprehensive information, which contains highest quality data. So again, if you want to have the best quality data, if you want to only stick to, if you're only looking at sequence data, then you must only take the curated fraction of the UniProt data, which is called UniProt/SwissProt. So if you just take that fraction of the data, then it is the highest quality data, which is more comprehensive. Of course, UniProt is currently the most comprehensive. Equivalent resource that is present for secondary information, secondary derived information is Interpro. So basically, if you do search in UniProt and Interpro, you can get most of the information. If you are looking for pathway-based information, then KEGG is the one to go. And if you're looking at protein-protein interaction, then string is the one to go. Okay, which is more up to date? Okay, most of these databases, which are like sitting at the root or the primary data node, are updated almost every day. Okay, almost every day, which is less redundant. You can apply a redundancy criteria yourself while you are doing the search. So most websites now incorporate such criteria. If it is not there, then of course you would have to download the sequences, apply your redundancy criteria, and then use that information to retrieve the data that you need. Which is more indexed? Of course, those databases that have been created by assimilating more databases are more indexed, because you can create more complex queries there. Again, UniProt and Interpro are the most common ones. There is also for those who are doing RNA related work or transcriptome related work. There is another browser that I have not covered here. It's called UCSC browser. Okay, so if you are doing a lot of RNA related work, then UCSC browser is the one that you should be looking at. That is most comprehensive for that purpose. Which web server responds most quickly? So these big web servers, they respond quickly because there are many mirror sites across continents. So I don't think this is an issue these days. And in such cases, when you have poor response, you can always change your mirror site and you can get the data from another mirror for quick download or abstraction of the data. Some important remarks. So databases may have many errors, especially when the annotations are automated. So not all databases are available on all servers. Update frequency is not same for all. So if you want to know how old or how new the data is, you should always look at the release notes. Okay. Cross references are always useful, and the more you have them, the less work you need to do at your end. So these are some tools that does meta search. So of these, only Entrance remains. So Entrance is hosted at NCBI. So other two have become obsolete. So you can go and look at Entrance in the NCBI website. Okay. This is one example of Entrance. So you can see it gets data from many different sources. Okay, and gives you. This is a snapshot from NCBI. So you can just go and look at it. And finally, I have come to the end of my talk. I showed you one website in the initial part of the lecture, bioinformatics.org, where people who have registered, their database links can be found. Okay, but if you look at this Wikipedia site, then some of the popular databases, you can go and look at. Okay, and since data is increasing, the question as to which is complete, which is best, and which is worst, all these are very relative, because what is good today will become bad tomorrow. What is bad today will become good tomorrow, and what is live today may be dead, and new database and new archivals will come up. So this will keep on going, and if you are interested in this area, you have to take some special interest to keep a tab on the new databases coming up. I think I'm done. So I think I finished on time. Yes, it's 7 o'clock. So thank you, everybody. Questions?
Any questions? Hello. This is to the participants. Any questions?
Sir, I have one question. Myself, Iqbal.
Yeah.
Am I audible, sir?
Yes, yes.
Sir, actually, in one slide, you said about image-based databases.
Yes.
So can you please elaborate on that? Actually, I have
Yeah, so
you have already shown an image containing some
right.
pattern.
Right. So this is images of a 2D gel. Yeah. A two-dimensional gel electrophoresis. So in molecular biology, you want to separate proteins from cells. Okay. So when you separate out proteins from cells, they come as some spots, and these spots make some patterns. Okay, so then what people have done, they have taken all these pictures of these gel images and they have archived it. Okay, they have used different experimental conditions and based on which different gel images have come. So these are now archived. So what people do is that they take these images and they analyze them.
Yeah, sir. That one actually I want to know from that. Which particular websites?
This is the Expressi. It's a Swiss Prot Expressi. So you
Okay,
can
sir.
go to Expressi, www.expressi.ch.
Okay,
Okay,
sir.
under that you will get these images.
Okay, sir. Thank you, sir.
Yeah. Okay. Anybody else? Okay, looks like there are no questions.
So if no more questions, any questions, any more questions?
Sir,
Dear
I have a
participants,
question.
yes.
Yes,
Sir,
yes, please.
sir, the software which you have told, Superpose version 1.0. So, sir, in that I can see many things below BioPearl, Emboss, Cluster W, all those I can download, sir, for the practical. Like I have seen multiple sequence alignment and all those we can do. So, phylogenetic analysis, all those we can do, sir, from over there.
Which program you're talking about?
Sir, actually many programs are there here. So, sir, what to download and
What is the task that you want to achieve? What is your question?
sir, actually here many are
No,
there like
the
bio.
question is which database you are searching. As you are mentioning the superpose, so the superpose server, what I have shown you in the morning, that will just give you the two protein. You can superpose one after another, and so you can get the structural similarity. That is one thing. They are asking you to upload of the two PDB code or the things. So from that they will do this thing. And in Superpost server, are you getting all these things? What are you saying?
Sir, actually below I can see many things. Actually, sir, that's what I was BioPearl like emboss in
Okay.
acknowledgement. I can
Okay.
see.
Can you share your screen? Can you share your screen where you are getting this thing? So it will be helpful for him to let you know. If you kindly share your screen, Prem, can you allow her to share her screen?
Yes, sir. Yes, sir. Yes, sir. Ma'am, please share your screen.
Sir, actually it is in my laptop and I am just actually I'm not able to do that. From actually mobile. Sir, actually below that superpose version, below in the acknowledgement, I can see like it uses VADR, BioPearl, WebMol. So, sir, we can download everything or according to the tasks we can do, right? That
No. Actually, what you are saying is that means the superpose is using the algorithm which is written by the BioPal program. Okay. So what are you saying is that when superpose is you are using, superpose is basically you are doing a program. But what is this thing? How it is, the which algorithm it is following? I think that these are acknowledgement means So they are acknowledging BioPerl because this program algorithm was written using the BioPerl. I think, Professor Paul, I am talking the right thing.
Okay, sir. Okay, sir. Thank
Yes,
you.
this
The confusion.
superpose website, when it is superposing the two PDB structures and giving you the output, it is in the back end, it is using the BioPerl module to achieve the task. Okay. So that is why it is telling you that, BioPerl. So
Okay, sir. Thank you.
I think the answer is. Okay, anybody else? Okay, so for tomorrow's session, somebody has suggested that I focus on NGS and RNA-seq data analysis. So my question is that these are heavy duty stuff. How many of you have prior knowledge in these areas? There is somebody, SG is there, who has sent the kindly enlighten the NGS and RNA-seq data analysis in tomorrow's session. So all of you have a background in in uh, genomics and transcriptomics. Where do I start? That's the question.
Professor Paul, as the nature of the, to my understanding, as there are all kind of people are there. So I think you can start from the basic, and it is not possible to tell the NGS or in one shot. So that's why I think the first session, if you give them a little bit of the big data, what is this, how this is applicable to proteomics, how this is applicable to genomics and metabolomics is there, why we are talking this. So if you give a brief idea about this, what are the big data in the biology, and then if you take it one or two, that is NGS or something in this, and also a few things about the AI as it is mentioned. So artificial intelligence, how the in agriculture or all other things, these are kind of biological tools people are using. So I think this way we can,
Okay.
because this is a mixed population.
Okay, I will try to keep two concepts, and so that it is easier for everybody to understand. And for those of you who are actually interested in pursuing, I will give out some detail which you can follow up. I think that would probably be a better way of doing things. Huh? Okay, so I think with that we will end.
Yeah, thank you, Professor Paul, for enlightening us, and we will wait for tomorrow. Tomorrow, I am requesting all the participants, please be here by 11, okay, so that we can start the session just on dot 11, okay?
Okay.
This
Good
is
night.
my humble request to you all. So till then, good night.
Good night.
Stay safe.